Okay, good morning. Thank you very much, Hendrik, for asking me to speak. He asked me to give more of a sort of high-level overview of precision diabetes medicine, which is what I intend to do. So hopefully we'll be able to cover a few high-level points relating to what's going on at the moment in this area and what the future might look like. So, and for those who aren't aware, so LUDC was awarded an 8-year award from the Strategic Science Foundation. I guess that was 2 years ago. Can't remember when it was. And the focus of the centre is on precision medicine now. So we have a mandate to research and invest in precision medicine. Many of you will know that already. So just a couple of plugs first of all. So one is for the ADA's Precision Medicine and Diabetes Initiative, which was started about a year ago, and that the web link is there. But you just Google ADA in precision medicine, you should find it. So that's an initiative that involves partnership with the EASD, NIDDK, the major diabetes drug companies to try to develop a consensus statement around precision diabetes medicine and looking at things like re-stratification and patient engagement, regulatory engagement, these kind of things. I'll come back to this at the end, but that's one exciting initiative that's underway at the moment. And the other is the Nordic Society for Precision Medicine, which we had a meeting here in Malmo for this time last year, almost exactly a year ago, in fact. And then the society was launched at a meeting at Decode in June this year, but that's now starting to take flight. So if you're interested in knowing what's going on in the Nordic countries in precision medicine, then take a look at that website. So the concept of precision medicine, I would argue, is not a new thing. This is a quote from Garold in the famous book Inborn Errors of Metabolism, but what he wrote was that even those idiosyncrasies with regards to drugs and articles of food, which are summed up by the proverbial saying that one man's meat is another man's poison, presumably have a chemical basis. And that, at least my interpretation of that, is to say that even before modern molecular phenotyping and genotyping technologies were available, there was a recognition that people's response to drugs and foods were probably regulated by features of our biology, which is at the very heart of the concept of precision medicine, as I'll explain in a moment. But what I would like to dispel at an early stage is to say that precision medicine is a different type of medicine to what is commonly practiced. It isn't. It's using conventional medical principles, but with different data. And that's, if you think about the practice of medicine, it should always involve some degree of stratification and personalization. That's the artistic aspect of medicine. That's where the patient and the clinician develop a relationship to understand the context and circumstances of the patient's disease, and then to tailor the therapeutic approach to those features. But what differs today is the availability of data, and those data conventionally for diabetes are these sorts of things: family history, lifestyle factors, and then other clinical features of dysmetabolism. But now we have the opportunity to engage many other types of data in the medical process. And that's conventionally, or some of it is conventionally, or becoming conventionally termed big data, particularly from electronic health records, but also obtained through other systems. And if you look at what's going on in the US at the moment with the integration of industries that were, historically have had no interest whatsoever in medicine, and those might include some of the companies like Google and Dell and Amazon and so forth, companies that have made their business in primarily non-medical sectors. They are now partnering with drug companies and with patient organizations to mobilize the massive amounts of data that they accrue for other purposes to channel that into for medical use. And so that's big data, if you like, and how that is all integrated and then translated into a form that can be used by patients and practitioners is at the very heart of the concept of precision medicine. And there are lots of different definitions of precision medicine. This is taking one of the, I would argue, most widely accepted definitions, which is from the Precision Medicines Initiative in the US. But it simply states that precision medicine is the ability to classify individuals into subpopulations that differ in their susceptibility to a particular disease, in the biology and or prognosis of those diseases they may develop, or in their response to a specific treatment. Preventive or therapeutic interventions can then be concentrated on those who will benefit, sparing expense and side effects from those who will not. There is a little bit of a distinction between the term precision medicine and personalized medicine. Certainly in the US, precision medicine is more commonly favored. The European Union is still using personalized medicine, as are some regional organizations. In Denmark, for example, personalized medicine is used by the Danish Innovations Fund, and Nordforsk uses personalized medicine. But if you wanted to make a distinction within semantics, then what you might say is that the concept of precision medicine isn't necessarily trying to get down to the person, to the individual, but to work within subpopulations that can be characterized and treated differently, and not necessarily treating every individual differently. And that type of medicine, of developing a medical device or a therapeutic option, is very rare if you think about it. You could argue for prosthetics, that's true. When you make a prosthetic for a patient that is designed for that individual's biomechanics and limb formation, but in sort of chemical medicine, that generally is unlikely to be true, that you get down to the individual level. And so in simple terms, it's moving from a situation where you have a somewhat one-size-fits-all approach to medicine to one where you stratify the population into subgroups that can be treated, hopefully in a way that minimizes exposure to unnecessary side effects and treatment failures. And that is probably the most basic and fundamental feature of how precision medicine might work. And that is already to some extent happening today with the use of genetics to identify contraindications to certain drugs. And as the availability of sequence level data in clinical records increases, which it will over time, there will be an automated process of flagging for a given patient a drug contraindication based on their genome sequence data. So that's one way that we can view precision medicine, to break it down into these subpopulations using biomarker technologies to guide therapeutic choices. But that's really only the sort of tip of the iceberg of how precision medicine might work. And this is so when if a company has a drug that they want to get approval from the FDA or the EMA for, they have to write what's called a context of use statement. And this describes the different contexts of uses that biomarkers might serve in the development of therapeutics. And I think this is actually really a great user's guide to how precision medicine can work with biomarker technologies. And as you can see, there's about 8 different categories of contexts where different biomarkers will play different roles, ranging from diagnostic. Clearly, if you take type 2 diabetes as an example, it's a disease of exclusion. The diagnosis is given after many other potential causes of chronically elevated glucose are ruled out, so it's disease of exclusion. And it's a diagnosis based on a consequence of a disease process, not on the disease itself. And so you might one day hope for a diagnostic in diabetes which targets the disease defect, which could be one of many things, to give a more specific, if you like, precise diagnostic option. And that's probably one of the earliest ways in which biomarker technologies might serve diabetes medicine in a more precise fashion. But there are monitoring. At the moment we use HbA1c to monitor progression in diabetes, but it may be that there are better biomarkers than HbA1c. Prediction, trying to determine who's likely to develop disease early long before the diagnostic label is applied, may give the option to prevent some of the consequences of diabetes, which are, of course, the major concerns, and so on and so forth, and clearly identifying subpopulations that are going to be better suited to different therapeutics is the sort of holy grail of this concept of biomarkers in precision medicine. Okay, and this is just to sort of to look at this a little bit differently, just to say that the application of those different types of biomarkers and the context may differ, or the time at which they're applied may differ in the life course of diabetes, some occurring long before the onset of disease. And if you think about, if you take the Whitehall 2 data, for example, and many of you may have seen the Lancet paper from Adam Tabak and colleagues, which shows that the departure in glycemic trajectories in those who are eventually diagnosed with diabetes probably begins from those who are not, about 20 years before diagnosis in the Whitehall 2 study. It's a very, very long period of gradual glycemic deterioration, which, of course, if you were able to identify those individuals 20 years before diagnosis, you would do so at a time where their beta cells are probably functioning perfectly normally, and rescue interventions might be possible without the loss of beta cell function, which would be clearly far preferable than having to deal with a patient who's already lost a large proportion of their beta cell function. So when those biomarkers are applied will differ depending on the context in which they're intended to be used. So I just wanted to sort of remind us, how severe the problem of diabetes is. So about 1 in 12 people globally have diabetes, and about half of all adults have either diabetes or pre-diabetes. Pre-diabetes is clearly a somewhat controversial concept because many people with pre-diabetes never progress to get diabetes, and it's not really known whether pre-diabetes is a disease state in and of its own right, or whether it's just a prelude to a disease state. So the therapeutic indication in pre-diabetes is unclear, and certainly the regulatory authorities do not have clear guidelines on for drug companies on whether pre-diabetes should be considered a state where therapies should be developed and applied. But nevertheless, there is a large proportion of people with pre-diabetes who will go on, about half of them within 5 to 8 years, to get diabetes. So it's clearly an at-risk population. So we have many, many people with diabetes globally. By the middle of this century, it will probably be somewhere in the region 600 million people worldwide, and many of those people will not know that they have diabetes, and that is clearly problematic because you can't prevent the consequences of disease if the person doesn't. Well, you can, but it's difficult to if the person doesn't know that they have it. And the majority of the people who will develop diabetes, the major global burden of the disease, will not be in Europe and in the US. It will be in the countries where the populations are growing rapidly and aging. And those countries are typically China, India, and to some extent Brazil. So there are regions of the world where, because of the demographics, we will have many more people with diabetes. But also because of the biologic features of those populations, we certainly understand that adipose deposition differs considerably between people of Southeast Asian ancestry compared with people of European ancestry, and that deposition is typically more central and around the heart and so forth, and so the biologic features of some populations will render them at higher risk of certain complications of diabetes and adverse lifestyle adaptations, which are of course driving these epidemics. And so understanding the biology and having the opportunity at a population scale to measure and monitor biologic features, and then to understand how therapeutic options will interact with those biological features, will be very important in the future. So why do we need, why should we change the way in which diabetes is prevented or managed? Well, this is a slide from the Diabetes Prevention Programme outcome study. Many of you will know that the DPP was the largest intensive lifestyle and metformin intervention that had been conducted. It probably still is actually within the context of a tightly controlled RCT. The trial ended on average at 3.2 years, but then an outcome study was bolted on to the back end of that, which is still ongoing, and it's now up to about 20 years. But what you can see is the incidence, the cumulative incidence curves within the three intervention arms of DPP, the placebo control at the top, the metformin in the middle, and the intensive lifestyle intervention at the bottom. And those curves have remained relatively parallel over time, but clearly at this point at 10 years, about half of the placebo group have developed diabetes. Now I should add that they were high risk at enrollment, so these people all had impaired glucose regulation, both their fasting and 2-hour glucoses were impaired, and they were overweight or obese, and this is an ethnically diverse study population. In any case, the DPP was heralded, and it's safe, or it's accurate to say that the DPP, when it was published and the results for the lifestyle intervention generated a lot of media publicity, that the paper which appeared in the New England Journal of Medicine came after another paper in the New England Journal of Medicine from the Finnish diabetes prevention study 9 months earlier, which doesn't get nearly as much attention as the DPP, perhaps because it was an American study. But interestingly, the result was identical for lifestyle intervention, which was a 58% risk reduction in the incidence of diabetes in those who received the lifestyle intervention, both in the Finnish study and the US study, relative to those who received the standard of care control intervention. And that generated a lot of hope and publicity around the concept of lifestyle medicine. And metformin, as you can see, was in between. It was a 32% risk reduction. So metformin did well, but not nearly as well as lifestyle. And I could stop there and say, well, how wonderful, the two major modalities for, frontline therapies for either the prevention or early management of diabetes are. But I'd like to suggest that actually they should rename the diabetes prevention program to the diabetes delay program. And the reason for that is that you can look at these data in a slightly less optimistic way and say, what's the degree to which the progression to diabetes was slowed down, because that's really what they achieved. And with lifestyle, it's about 36 months delay, and with metformin, it's a 12 months delay. And the question then is that good enough? And of course the answer is no, it's not good enough, because that although that will convey some benefit in terms of complications later on, it's not going to stop the complications if you can't stop the disease. So it's not a truly disruptive, neither of these were disruptive interventions that just stopped the disease process, they just delayed the onset of disease. So that's really the sort of, I think, presiding argument for why we need better therapeutic and preventive approaches in diabetes, and that brings us back to precision medicine. I'm sorry this is a bit fuzzy, but the point of showing this is really just to get everyone on the same page in terms of the drug discovery pipeline or molecule to market pipeline for drugs, which it's an old paper, it's 15 years old, but in any case the process of the phase 1 to 3 clinical trials and having preclinical testing and so forth remains more or less the same within both EMA's and FDA's expectations. And that process takes, in the US, rather a long time and costs a lot of money. So it costs about $2.6 billion to get a molecule to market, and it takes 11 to 14 years. So this is a very high-risk game, to the extent that only 1 in 10,000 molecules eventually makes it to market. So there is a huge process of failure throughout drug development pipelines, and that gives you a conditional probability of getting a drug to market of less than 1%. So this is a very high-risk business, and the problem is it means that drug companies are risk-averse these days. They don't want to take too many risks in pushing molecules through this process because it can be incredibly expensive, and therefore if they have a drug that works, they will stick with it, and this crushes innovation. So adding to this, and this is largely due to FDA's increased expectations in terms of burden of evidence and the minimalization of risk and so forth, but the cost of developing drugs is going up. And so the whole dynamic that surrounds the concept of drug discovery is becoming more challenging. That environment is more challenging. And even when you have got drugs through that process, many of them don't work. None of these are diabetes drugs, but this simply illustrates the extent to which the major blockbuster drugs don't work. Okay, and you can discuss the reasons why they don't work, but nevertheless, up to about 90% of people who take some of these blockbuster drugs will not benefit from, depending on the drug, will not benefit from the drug. Either they don't take it because they're intolerant to it, or the drug simply doesn't work. So there is a major problem with both the process of drug development and discovery and the availability of effective drugs in the marketplace. So things have to change. And one of the things that offers a glimmer of hope is the use of genetics in drug development. And this is, I put this schematic on there just to describe the process in which genetics is sometimes applied to the drug discovery process. But the statistic that's worth keeping in mind is the extent to which, if you have you have a genetic mutation that perturbs a pathway, in other words, provides evidence of mechanism within that pathway, and that perturbation can be targeted with a drug, that is a very useful way to improve the success of drug development. It doubles the success rate roughly. So that is why, for example, Decode were able to sell access to their human genetics database that covers the entire Icelandic population to Amgen for a staggering $450 million. They didn't give the database to them for that, they just allowed them managed access to the database. So that tells you something about how valuable genetics is in the development of drugs. And Decode is one of many initiatives now that are developing these massive genomic resources that are being linked with comprehensive phenotypic databases worldwide. This is not an entirely inclusive map of everything that's going on globally, but it tells you something about the enormous amounts of resources that are being pumped in to precision medicines initiatives and databases that can be used for precision medicine globally. And that, in Europe, involves, one aspect of what's going on in Europe is the Innovative Medicines Initiative in the EU. The EU have pumped in about 1.3 billion euros into the IMI projects. There are now more than 100 projects, IMI projects. Several of those are diabetes related, and most of those that are diabetes related are coordinated or have a strong steer from LUDC. So we've been very successful at LUDC in designing and managing IMI projects related to diabetes. Leif has led Summit and co-leads Rhapsody, and I'm involved in Direct and Rhapsody, and Maria Gomez leads Beat DKD. So there are all of these IMI projects going on, which have been a hotbed for collaboration between industry and academia, which apparently is a very good thing, because about 50% of the most clinically impactful drugs, I don't know how that's defined in the paper, but have been discovered through private-public partnerships between academia and industry. So that's a good thing. And in the US, the sort of analog is the Accelerating Medicines Partnership, which also involves collaborations between industry and academia. And they've been very effective in building large databases. So the Million Veterans program, which is run through the Veterans Affairs Hospitals, is enrolling 1 million patients into a database that will be used for these private-public partnerships. So I mentioned Direct. This is a project that we've run in collaboration with 30 other partners, academic partners across Europe, and involves 7 clinical study centres, one of which is here. There's a Malmö and Lund study centre that Magnus has overseen in the last few years, where we've brought new patients in and generated new cohorts. And we've just finished data collection in this after 7 years of this process. So we recruited about 3,000 people. 2,200 came from existing prospective cohort studies, including the Malmo diet and cancer study and several others, identified because those individuals were deemed to be at high risk of glycemic deterioration, which is subtly different from saying they're at high risk of diabetes. And then the other 800 or so individuals were recruited through diabetes registries, and this was the source of recruitment for the Malmo Lund centres, where we identified people who had recently been diagnosed with diabetes and invited them to participate in the study. Now, direct cost almost 50 million euros, and these two studies cost something like 20 million euros, so it's expensive to do this. But this gives you an idea of the intensity with which these participants were studied. So we, as I said, had the opportunity to enroll participants in the pre-diabetes cohort from existing cohort studies, and so we had information on those individuals that dated back maybe a decade that allowed us to look at their changes in glycemia over time, their body mass index, and so forth. So by the time they were enrolled into Direct, we knew quite a lot about them. And then they had, sorry, the T's been lopped off there, but they had 3 time points where they were intensively studied. We had this parallel diabetes cohort, which essentially we, to the extent that we could, ran an identical protocol so that we had these 2 cohorts that could be compared. The differences being that instead of doing repeated HbA1cs, we did urinary C-peptides, and instead of doing an FSO-GTT, giving a 75-gram glucose load, we gave them a mixed meal drink because they had diabetes. But apart from that, they all underwent a number of sort of phenotyping approaches. They had MRI scans of the abdomen, heart, liver, pancreas, and so forth. They did 10 days of triaxial accelerometry, we did diet records, and we collected stool samples and measured these participants. And then from that, we generated a lot of data around genetics, RNA-seq, microRNAs, proteomics, metabolomics, and then microbiomics. So we have this very comprehensive deep phenotype database in these 3,000 individuals. And that database will be made accessible through managed access to anybody who has a good research idea as of next summer. So that's a lot of work to collect all this data, but it will be put out into the to the research community for access within about 6 months, once all the final data is cleaned and accessible. Now, in terms of the opportunity to practice precision medicine in complex diabetes, let's call it type 2 diabetes, people often look to cancer as the poster child for where precision medicine has worked very well. And on the one hand, that's certainly true that what's been learnt around the somatic mutations that cause cancer, and then drugs that have been designed to affect those somatic mutations in a favourable fashion, has led to a process of diagnosis that involves genome sequencing, coupled with the application of a targeted drug therapy to address that genetic insult. And that, in some cases, has been incredibly successful, and some of the most aggressive cancers have been stopped and reversed using this approach. But unfortunately, cancer and pretty much every other disease are very different in the fact that from a genetics point of view, we're not talking about somatic mutations as we understand it at this point, at least in complex metabolic diseases. We're talking about germline DNA variation, which conveys a different or requires a different approach if you're going to use that in medicine. There are, of course, examples in monogenic diabetes and with permanent neonatal diabetes and some types of maturity onset diabetes of the young. Sulfonylureas have been proven to be effective in people who carry mutations that affect the gating of the the potassium channels and the depolarization of the beta cell membrane. And this is just a schematic that illustrates what happens here, where you have you understand the beta cell dynamics here in terms of calcium and glucose, and then insulin secretion. But when the calcium channels are mutated, so SIR1 and KER6.2 mutations are the ones that were initially identified to cause this problem. The membrane remains hyperpolarized, and if it's hyperpolarized, the insulin, the exocytosis doesn't work, and so sulfonylureas fix this problem and allow insulin to be released, which is a wonderful story. I mean, many of you will know what this means in terms of having had patients who were often misdiagnosed as being type 1 diabetics and insulin treated, having the capacity to take them off insulin and give them a sulfonylurea tablet, allowing them to secrete their own insulin. So it's really a wonderful story about how genetics and drug therapy in diabetes have worked very well. But that's not really translatable to type 2 diabetes, where you have very complex processes, many of them not being genetic, and if they are genetic, they're probably polygenic, which means that there are lots of small genetic effects working in concert to cause very different types of diabetes that we call type 2. So this is probably not translatable either. So what, from a genetics point of view, what's been done recently, which is, I think, a nice step forwards, a meaningful step forwards in the use of DNA variation in the classification, subclassification of type 2 diabetes, is something called the development of process-based risk scores, which Mark McCarthy and Anuba Mahajan and others developed in Oxford, which is reported in this paper earlier this year in Nature Genetics. And what they did was use the process of clustering to take, and I should say there are now just over 400 relatively independent genome-wide significant loci for type 2 diabetes. This is not using all 400, this was only using about 180, but the process is the same. So what they used was a sort of conditional probability approach to cluster the genetic variants into groups of gene variants that also act upon pathways and processes that precede the onset of diabetes, and they are things like impaired lipid metabolism, insulin secretion, insulin action, and so forth, and development of adiposity. And doing that provides some potential to subclassify the population into these subgroups that may benefit from different modes of diabetes therapy. It would be remiss of me if I did not mention that there's also a phenotypic clustering approach that's been pioneered by Leif and Emma and others, and there are others who've also used other genetic clustering approaches. This is a paper from Miriam Adler and Jose Flores at MGH. But the principle is simple, that if you were able to subclassify people with diabetes into subgroups of different etiologies, we already know that different drugs act on different organs and tissues in different ways, and so that subclassification very rapidly could lead to the reallocation of therapeutic options in diabetes. The problem is, and of course there's always a problem, is that this is a highly probabilistic scenario, and at this stage the ability to cluster an individual into one of these subclasses of type 2 diabetes is not an absolute distinctive way of operating, and the better way to say that is a person will have a probability of being in one or more clusters, and therefore choosing a distinct therapeutic pathway is not simple, because it's actually one plus maybe a bit of others, and that hasn't been worked out yet. I also wanted to stress that, it's not all about pharmacotherapy. Some of you will know that my interest and passion is actually more to do with lifestyle medicine than drug therapy. But it is emphasized by in this NIH statement around precision medicine that lifestyle and the environment are important too. So we probably should keep that in mind. Of course, there are examples like phenylketonuria, where gene screening at birth leads to, in some individuals who carry the mutations, recommendations about diet that are low in phenylalanine. That's a monogenic example, and there are many other monogenic examples that exist, including lactase persistence, fatty acid desaturization in the Greenlandic Inuit, gluten intolerance to cereal crops, and so on and so forth. Most of these are probably not relevant to the population at large. These are relatively rare, often from isolated population examples. But it does stand to reason that if you think about the external environment, the food we eat, the things we're exposed to, at some point, for any environmental exposure to impact our phenotype, whether it's a disease or health phenotype, it doesn't matter. It involves the genome. You have to have the genome involved to develop proteins and products. And therefore, if there's variation in the central dogma, in the process that goes from gene transcription and translation through to the formation of small molecules and so forth, that variation in these pathways, whether it's at a nuclear DNA level or at an epigenetic level or others, that will affect the processing of different nutrients and molecules that we put into our bodies, and in terms of how it then conveys itself in a phenotype. So this concept of interaction between the environment and our biology stands to reason, and it's not a sort of science fiction thing. It's actually very eloquently illustrated in a number of studies now, and this is one from the Weizmann Institute in Israel, from Erin Segal's lab. And what they did here was took a cohort of 800 young adults, and they gave them set meals for a week. And during that week, they monitored their glucose continuously, they collected stool samples and measured physical activity and monitored diet and so on and so forth. And this just shows you the CGM trace looking at the postprandial glycemic response to food. But one of the things that they illustrated in this paper very nicely is that an individual's glycemic response to food differs dramatically from, or can differ dramatically from one person to the next. So this shows for bananas, they picked out one individual where there's a glycemic peak when they eat bananas, which is what you'd expect, particularly if it's a browner banana, which is more oxidized. There's quite a lot of sugar in bananas. But when this person ate a cookie, I don't think it was a blue cookie, but a cookie, they did not get this glycemic response, whereas another participant had the contrasting glycemic response to these two types of food. Now, that there are lots of crappy studies out there that have sort of suggested this, and of course this could just be random sort of stochastic variation in glycemic response. But what they also showed is that these patterns of glycemic response are sometimes repeatable with an individual, and when they're repeatable it starts to get interesting because it starts to tell it's not just by chance. So they also did a study on glucose and bread illustrating the same sort of thing. But they then went on having ascertained a lot of biomarker information in these individuals, particularly around the metagenome in the gut microbiome, and they predicted the ideal diet for individuals, and then they randomized people to the best diet and the worst diet they could be given. They demonstrated they could predict glycemic response by manipulating the diet, which is really a landmark study in that respect. This is just a quick example from work that we've done with the diabetes prevention program that I spoke about earlier, and the Look Ahead trial. Look Ahead is a study that's very similar to the DPP in concept and design. The difference really is that the DPP focused on people at risk of diabetes and tried to prevent the onset of disease, whereas Look Ahead started with people who already had diabetes and tried to prevent the onset of complications in those individuals. But the design of the two studies, and indeed the study centres and investigators, was heavily overlapping. So they're quite nice studies to work, to put together into combined analyses. So what we did here was we took at that point, the 100 or so known obesity loci, and we looked to see whether any of these loci influenced the response to interventions in terms of weight change. This is broken down for simplicity. It's not a magic trick. I'm not trying to get something by you. I'm just breaking it down to make it easy to describe. I'll tell you now that for this particular variant at this MTIF3 locus, all of these variants were already firmly established to be associated with heavier BMI. So the G allele conveys risk of being obese. That's the characteristic of this genotype. The A allele is the reciprocal genotype or allele. When you control for baseline weight in the trial and you look only at men who were randomized to the standard of care control intervention, there's a subtly heavier weight in those individuals across the 4 years of the intervention period. But if you look at men who receive the lifestyle intervention, the most notable thing, of course, is that the nadir of the weight loss trajectory is at about 1 year, and that's common with lifestyle interventions. Most people lose most of the weight by 1 year and then start to regain it. But what's intriguing is that the obesogenic allele actually conveys more weight loss than the non-obesogenic allele, which is surprising, but also translates to a gene by intervention interaction. And you see the same thing in women, and you see the same thing in look-ahead. So this is probably real. And to put this another way, what does this really mean? Is it clinically relevant? Well, it means that the delay in weight regain in pre-diabetes, individuals with pre-diabetes, is about 18 months if you carry both copies of this allele, and in people with diabetes, it's about 9 months. And this is a fairly large, it's over 2 kilograms difference in absolute body weight at 1 year. So it's probably a clinically meaningful amount of body weight loss with intervention. And interestingly, we had nothing to do with this. This was published almost at the same time, a couple of years ago. But this is looking at bariatric surgery. They did basically the same thing as we did, but for bariatric surgery. And what they found was their top hit is this SNP, which is in high linkage disequilibrium with the one we'd looked at. And they showed that variant also is beneficial for weight loss retention after bariatric surgery. So why, we don't know. So just to wrap the talk up then, this is going back now to the ADA Precision Diabetes Medicines Initiative, and this is a pipeline or a process illustration of at least what we think within the ADA initiative we should be working with. Most of what I do, and probably most of what the researchers in this room do, is at this end of this pipeline around discovery and interpretation of data and all the cool stuff that gets published in high-impact journals. But there isn't really that much going on around this end in precision diabetes medicine at the moment. There are no health economic assessments published relating to precision medicine in type 2 diabetes at this time. There are a couple in MODI, but nothing in type 2 diabetes. So there are big gaps here. And obviously there are issues like clinician education and patient engagement that are pretty poorly done at this stage, perhaps justifiably. And also regulator engagement, having conversations with the EMA about processes that might lead to developing new drugs or approaches. So there's a lot of work to be done here, and this is probably only a small fraction of the things that needs to be done. Do I have 2 more minutes? Yeah. So these are the bonus slides. This is a Christmas special, but this is just not really so much to do with precision medicine, but this is. So we did I was going to say a small exercise. It wasn't. It took a year or something to mine all the data to do this properly. But this looks at the how genetics research has been done over the GWAS era, the last 12 years or so. And what this shows is the extent to which data has come from the Nordic countries. So this is a plug for the Nordic region for at least population genetics, which is one key aspect of precision medicine. And what you can see is that for type 2 diabetes, coronary artery disease, schizophrenia, and lipid disorders, large chunks of the data that have been used in these major GWAS papers, these are the Nature Science, Nature Genetics papers, has come from the Nordic countries. Indeed, probably about a quarter of all the data that's gone into all these papers has come from the Nordic countries. And this is going now specifically to diabetes. And the sad thing is that if you look at the distribution of first authors and last authors, even though the US has not contributed greatly in terms of data over the years, the authorships are really driven by the US and to a lesser extent the UK. And you can see Sweden, which has been one of the major contributors through LAIF in terms of data, is getting a very small slice of the cake in terms of authorships on papers. And this is not to say that this is necessarily unfair, because it may be that, and it probably is, that the resources for genotyping and also the sort of intellectual resources for bioinformatic machinery has been, you know, at places like the Broad Institute in Michigan in the US, and so they've been really driving that aspect of it. But it's just to say that probably we should try to affect this in the future. We have great data in this region for genetics and precision medicine probably, but we need to get in the driving seat. And this is just now looking at the gender distribution in these diabetes. I should say this is about 35, the top 35 diabetes genetics papers published over the last decade or so are assimilated into this analysis. And what you find is that the cohorts are fairly evenly distributed in terms of males and females. And in fact, the distribution of first authorships is in favour of females, and so clearly a lot of the new wave of data analysts are probably female, because that's generally what first authorship reflects. But there aren't so many senior authors publishing on diabetes genetics papers in the published literature, so that may just be a legacy effect of how science has been done, and probably how the Western world has operated for that matter. But hopefully this will translate to a more balanced gender distribution in terms of men and women driving diabetes genetics in the future. Okay, and that's it. Thanks.